Hi guys, and welcome to the review study session. We're going to go through the study guide, so if you guys want to have it open alongside, then I recommend watching this video. I would say if you haven't completed the study guide yet, pause this video. You want to try and answer all the questions on your own first, just because that tells your brain, hey, this is important stuff. That's why I'm trying to retrieve the information so I can answer the questions. And then after you've done that, then watch this video. Use it more so to correct, to make sure you have the right answers rather than to get the answers in the first place, if that makes sense. So if you have everything answered, then great, we can go ahead and jump in. If you decide to use this to answer all the questions, then you're welcome to do that. But like I said, I would caution answering the questions first, then using this video to check to make sure you got the right answer. So I've got my pen here. We're going to just kind of go through the review kind of question by question. So starting with the first question from Unit 1, which was how to science, what are the 6 steps of the scientific method? So you can find this information in the slides from the first few weeks of school. So there's 6 steps, 1, 2, 3, 4, 5, 6. So to remember these 6 steps, what you really want to do is instead of just trying to memorize, you want to instead think about how you do an experiment in the first place. We've done a few experiments in class, and so I would say if you, instead of just trying to memorize the steps, refer back to when you did an experiment, which steps did you do, and that'll actually make it a lot easier for you to then remember those 6 steps. So I'm pulling them up right now here. Let's see. So the first thing that you want to do when you're doing an experiment is you want to figure out what you're doing your experiment on in the first place, right? So the first way that you do that is you observe. You have to look around. You have to see what's going on in the world. So, for example, when we were doing the diffusion lab, you had to create your own diffusion experiment by making observations. You observed what happened in the demonstration, and then you use that information to then create your lab. So remember, you want to make observations. Well, after you've made your observations, then you want to figure out what you're testing, what you think is going to happen, what you're looking to do. So that's where forming a hypothesis comes in, right? So when you were making your observations about the diffusion lab, you're like, okay, great. The hot water seems to be diffusing faster than the cold water. So then your hypothesis for the one that you created could be, well, if I'm doing an experiment on really, really cold water, really, really hot water, based off of my observations, my hypothesis would say if I have hot water and cold water, then the diffusion will be faster than the hot water than the cold water, because that's your hypothesis. You're forming a hypothesis made off of your observations. So form a hypothesis. This is your educated guess of what you think is going to happen. Then you have to design your experiment. So what are you going to be doing for your experiment? Well, how am I testing this? What do I think is going to happen? And how do I design an experiment that allows me to measure that? To design an experiment. Then, after you've designed your controlled experiment, you'll do the experiment. And when you're doing the experiment, you're collecting and analyzing data. And remember, the data is the information that you get. That's what tells you about what you're seeing. Then, now that you have your data, now that you have the information, you can draw a conclusion. And say, okay, well, if we're going back to the example of the diffusion lab, if your hypothesis was that the hot water would diffuse faster than the cold water, and you designed your experiment with cold water and hot water, you collected your data, which was the time it took for it to diffuse. Now you're drawing a conclusion. Well, if it was faster diffusing in the hot water, then that would be your conclusion, that in fact the temperature did increase the rate of diffusion. The hot water did have a faster diffusion than the cold water. And then communicating results is where you then take that information, you say, cool, the results showed me that the hot water diffused faster than the cold water. I'm going to communicate those results. I'll put it in a scientific journal maybe. I'll write it on my lab report, and then I'll share with everybody that this is what happens. So these are those 6 steps. Remember, we've done these in class, and so it's not so much about just memorizing what these steps are, but instead going through the experiments that we've done and remembering when we made observations, when we formed hypotheses, when we designed experiments, collected and analyzed data, drew a conclusion, and communicated results. So that is the first question here. Let's go to the next question. In terms of an experiment, do we directly change the independent, dependent, or control variable? So when we have our experiment, remember we said we wanted to design a controlled experiment. And so to have a controlled experiment, we need to be aware of what our variables are. So we'll do independent. We'll do the definition here. Dependent. And control. So the independent variable is the one that we directly change to answer the question. So we directly change the one that we decide So for the independent variable, I'll put variables up here. For the independent variable, we directly change it and then we decide what happens. So we decide if it's going to be hot water or cold water. We decide if it's going to have 3 ice cubes or 4. We decide what temperature it's going to be. If you were doing something that wasn't temperature related, then you'll decide the other factors, right? So you're controlling the independent variable because the variable is independent to the rest of your lab, right? Because you're the one controlling it. It doesn't really rely on anything. So then, as the opposite, the dependent variable depends or changes responding to the independent variable. So it's the part that responds to what we change. It depends on the independent variable. It depends on what we're doing, right? It depends on if we want to do hot water or cold water. It depends on the temperature. So that's the dependent variable. And finally, the control is what stays the same in every trial. So this would be, for example, if you're doing hot water versus cold water, well, you want it to be the same temperature, hot and cold, right? You don't want some hot to be like 90 degrees and other hot to be 100 degrees unless that's the variable that you're measuring. So I guess another example would be like you want it to be hot water, you want it to be the same amount of water, you want it to be the same kind of water, you want it to be just the same. You want it to be consistent through every single trial. That way you know that your results, the way that your dependent variable changes, isn't based off of anything except for your independent variables, the ones that you directly want to control. That's the whole point of having then the control, because then that helps you know that the results that you see aren't because of all these extra things, they're just because of the independent variable. So that control, once again, is what stays the same in every trial. And then let's see, for the last 2 questions here, I ask about quantitative and qualitative data. And so if you're collecting and analyzing data, that's where we'll have those 2 forms, qualitative or quantitative. So quantitative is quantity. It's amount. How much? Measurement. If you're doing the diffusion experiment, then your quantitative data is the quantity of time it took for diffusion to occur. How long, right? That measurement. Qualitative is measuring quality. And so these are like traits and descriptions. So you could say, for example, if you were looking at a flower petal, quantitative would be how many flower petals there are. There's 5 flower petals. Qualitative would say, and they're yellow or they're orange. It's the description of what it looks like, how it appears. What are you observing? So qualitative is the quality of it, the different kinds of characteristics that we notice, that we can describe. And then quantitative is what we can count, the quantity, the measurement. So this is essentially a summary of the first unit, How to Science. So that's that for you. Let me step out of the frame. If you guys want to screenshot and then go back to it, I would recommend doing that as a good study tool. The next section is ecology. So what is an ecosystem? Going right from the beginning here. So an ecosystem is all the abiotic and biotic members or factors of a biome, or like kind of just an area of focus, where you're looking at, where you're studying. So that is ecosystem. And now the next question are what would be abiotic and biotic. So if we remember, abiotic is without life and biotic is with life. So if you remember, like the A meaning without, so that's why bio being the study of life, biology, bio, so that's biotic is alive, and abiotic is not alive. So what would be abiotic? What are some examples? Those would be things that aren't living, like sunlight. The sun's not alive. Well, technically speaking. Sunlight, you could say wind, you could say like air, water, anything that's on periodic table, for example, any of those kinds of chemicals that we might need, like rocks, minerals, anything that isn't alive. So here's some examples. It's more than 3. So then biotic would be kind of everything that's alive. So you'd say animals, plants, bugs, bacteria, algae. The list goes on and on, right? There's tons of different things. So if you were looking at one specific ecosystem, for example, let's say a tide pool. Tide pools are really cool. I love being able to visit them. There's so many in San Diego. And they're so great to see little examples of life, little mini ecosystems in each tide pool. So let's say an abiotic factor of a tide pool would be sunlight. It would be like how much ocean water, or I guess seawater, they get access to, how like dry it is, or wet it is. So those would be the abiotic, right? And then the biotic would be what kind of animals live there. So are there sea anemones? Are there crabs? Are there fish? Is there maybe a sea lion, like in the La Jolla Cliffs, that decides that they want to hang out on the tide pool? So lots of different biotic factors and lots of different abiotic factors. And remember, those work together to create an ecosystem and all of the different conditions of it. So those are kind of the main parts of an ecosystem that we want to look at. And then another part of interest that we learned about in ecology are then how those biotic factors interact. What are those relationships? So if we have the 6 different ecological relationships, there's 6 of them. And we did PowerPoint presentations. Each of you guys did one on at least one of the 6. So hopefully you're familiar with at least one of them. And then you'll have a chance to kind of review over the others. So we've got mutualism, commensalism, parasitism, predation, interspecific competition, and intra-specific competition. So, looking at these 6, hopefully, like I said, one of these at least sounds familiar to you. And these are really some of my favorite parts of ecology because I love to see how species interact with each other. I love to see how they evolve with each other. Let's go ahead and go through each of these specifically. So mutualism is plus plus, both benefit. And so an example might be like the bees and flowers. So bees and flowers. Bees get to benefit because they get food when they pollinate. Flowers get to benefit because they're pollinated, which means they get to continue on their life. They get to continue on their generations, right? They get to share their genetic information. So bees get food, flowers get pollination, which is really important. So that's mutualism, both benefit. Commensalism is where one benefits and the other doesn't really experience anything. So one example are remoras and sharks. Remoras benefit because they get to swim in the slipstream of the shark, so they kind of get pulled along and they get to eat any leftovers that the sharks leave behind. They're essentially like a dog that's waiting underneath the table for any scraps that get kind of tossed off the table. Meanwhile, sharks don't really experience anything because there's just a fish following them around. And that's kind of it, right? They don't really experience anything. Parasitism is a negative positive. So you have the parasite. We'll say, for an example, we'll do tapeworm because these are pretty common. Tapeworm and humans. So tapeworms benefit because they eat all of the food that we eat. They get all the nutrition from everything that we digest. Humans, on the other hand, suffer because our nutrition is getting taken away. The food that we eat is literally being sucked out of our body to feed the tapeworm. So parasitism is a negative and a positive. Parasite benefits, the one that's being parasitized on is suffering. Predation is positive, negative. For example, if you have a lion and a zebra, well, the lion's benefiting because, hey, it's dinner time. The zebra's not benefiting because, hey, I have to be your dinner, which means I died. So that's predation. Interspecific competition is when you have competition between two different species. So interspecific is negative because they're both competing against each other, and this is between the different species. So this would be like two species fighting over the same area to try and live, like in a tide pool. If the sea anemones and the barnacles want to live on the same rock, then they're competing with each other. That's interspecific competition. Intraspecific competition is also negative because they're still competing, but this is competition between the same species. So this would be like two reindeer. Two male reindeers are fighting against each other for access for all the females. So they're competing against each other. They're two of the same species. Or kangaroo. They're fighting against each other once again, just trying to get access to the females. So interspecific, remember, is between different species. Intraspecific is between the same species. I think of it because the letter A is in the word same. That's kind of the best I got for you guys. So that's ecology. Kind of the main ideas that I'd like you guys to carry on about ecology for the final here. So then let's see. Let's look at the next one. That's where we're going to get into what we touched on a little bit more recently. That's where we'll talk about the chemistry of life, which goes into like atoms. I know that the dreaded radioactive isotopes, all of that stuff. So let's see. This first question here are what particles are found in an atom. What can you learn about an element when looking up in a periodic table? Draw an atom for carbon. I'm just going to do all of these by showing you the example of carbon. So let's draw an atom for carbon, which has an atomic mass of 12 and an atomic number of 6. So the atomic number tells us the number of protons. So that's one of the particles found in an atom. It also tells us the number of electrons, which is the second type of particle found in an atom. And then finally, we have the atomic mass, which is number of protons plus the number of neutrons. And neutrons are the third kind of particle found in an atom. So then to find the number of neutrons, you have to subtract the number of protons, which is the atomic number. And so if we need to draw the atom, then we'll just subtract here. That tells us we have 6 neutrons. And then what else can you learn about an atom when looking it up on the periodic table? Well, we just learned the number of protons, the number of electrons, and the number of neutrons. What else can you tell? Well, you can tell what atom it is. If you look at the color of it, sometimes there's a legend that will tell you that carbon is a nonmetal. You can see its atomic weight, all of that stuff. So that's some of the information you can get while looking it up on a periodic table. And then finally, let's draw it. Let's see what it looks like as an actual atom drawing. So now that we have this information, we know it has 6 neutrons, 6 protons, and 6 electrons. The last piece of information we need to know is where do these go in the atom? So both their neutrons and protons go in the nucleus. Go in the nucleus. Your electrons go in the orbitals. Remember, the first orbital can hold 2 electrons, and then anything that's not the first orbital can hold 8. So then we'll do all others can hold 8 electrons. So knowing that we've got 6, 6, and 6, let's go ahead and draw it. So we'll do 6 neutrons, 6 protons. Those are right here in the nucleus. And then our first orbital has 2 electrons. And then my second orbital has 4, because 2 plus 4 equals 6. My first orbital can only hold 2, so that means the other 4 go on the outside. And so this is my nucleus with my 6 neutrons and 6 protons. These are my 2 orbitals with my 6 electrons, and that is my carbon atom. So that kind of helps answer the first 3 questions, and then we can kind of then switch gears and head over to scientific notation, isotopes, all that stuff I know we had a lot of fun with. Alrighty, so let's get into scientific notation. I know this normally looks a little bit more intimidating than it actually is because all the information that you need is right in the question itself. So let's start with this one, 1.2 times 10 to the negative 1. So we see it's negative, which means that we're going to be moving to the left, and it's a 1, so we're going to be moving to the left 1 time. So take that decimal point, move it to the left, and our number is 0.12. So then let's go to the next one, 7.62 times 10 to the 3. So this one's positive, which means we're moving it to the right, and we're going to move it 3 times, 1, 2, 3. If there's no number there, you'll just add a 0, so it's 7620, 7,620. So that's that. That's pretty much scientific notation, easy peasy, so easy to come up with examples as well. So you can just change the exponents, and that'll give you practice to move those numbers and decimals around. So that's that one. Then let's get to the isotope radioactive decay, because I know that's a little bit more of a difficult one for sure. So let's see. If I have 200 isotopes of chemical X, which has a half-life of 2 hours, how many isotopes will I have after 6 hours? So how many after 6 hours? So remember, the first thing that you want to do is to figure out how many half-lives have occurred. So that's where you take the total amount of time. So total time, in this case, is 6. And then you want to divide it by the length of a half-life, because you want to find how many parts of a whole occurred. So you're going to do our half-life, which is 2. So that tells us that 3 half-lives have occurred over 6 hours. So another way to look at it is if I have a timeline, if this is 0 and this is 6, and we go 1, 2, 3, 4, 5, I want to know how many half-lives occurred. If a half-life is 2 hours, then this is a half-life, this is a half-life, and this is a half-life. So from 0 to 2 is 1 half-life, from 2 to 4 is another half-life, and from 4 to 6, that's another half-life, which is also 3. So it's really just asking how many chunks of time equal a half-life, and how many half-lives then are in the total amount of time. So that's the first step, is to figure out how many half-lives have occurred. That's step 1. Now step 2 is to then cut the number of isotopes in half that many times. So because we have 3 half-lives, that means that we're going to have 200 times 1/2 times 1/2 times 1/2. Because we're going to cut 200 in half 3 times. So what's 200 in half the first time? So over the second half-life, we have 100, right? Because that's 200 divided by 2 equals 100. And then 100 divided by 2 equals 50. And then 50 divided by 2 equals 25, which means that at the end of the third half-life, which is the same way of saying at the end of the 6th hour, we have 25 isotopes of chemical X. So that's how you would solve that one there. Okay, then let's see. What is an isotope? So after all of this, what is an isotope in itself? An isotope is just a version of that element. It's an atom of a certain element that has a different number of neutrons than the more stable version. So let's go back to carbon. Carbon has, normally in the stable form, we'll do stable and then we'll do isotope. In a stable form, it has 6 protons, 6 electrons, and 6 neutrons. An isotope of carbon, for example, carbon-14, have 6 protons, 6 electrons, and then might have 8 neutrons. Remember, we can just tell by subtracting the atomic number by the atomic mass, or from the atomic mass. That's how we find the number of neutrons. So an isotope is, once again, it's just an atom of a certain element that has a different number of neutrons than normal. And you can have more or less. So let's do another example. Let's do carbon-10 isotope. 6 protons, 6 electrons, 4 neutrons. So this is an isotope, and this is an isotope. And this is the stable, most naturally occurring, most common version. So once again, just isotopes are just atoms of an element that have different number of neutrons. So then that's how you'll know if you have an isotope, is just by counting the number of neutrons. And if they're the same as the stable version that's reported on the periodic table, well, then you know you have the regular stable version. If it's different than that number, then you have an isotope. It's pretty easy. Okay, so then let's kind of switch gears a little bit to macromolecules. What is a macromolecule? What do they do for our body? So I'm just going to make a table here of all the different kinds. So we have carbohydrates, proteins, lipids, and nucleic acids. Nucleic acids. So what is the main function? What do we do with these? So carbohydrates are instant energy. They provide us with quick, easy forms of energy. Eventually they turn into ATP, which, as we now know, ATP is kind of like the money that our cell uses, that form of energy that we can transform to be able to do certain things that our cell needs to do. They can also be used as structure or support, such as plants use carbohydrates. Bugs will use them as an exoskeleton, so for protection. And examples include like sugar, glucose, sucrose, anything that ends in an ose normally is a carbohydrate because it's a sugar. So we could do like candy, ice cream, also fruits and vegetables, pasta, bread. Those are all different kinds of carbohydrates. Sorry, it gets a little messy down here. Fruits, veggies, pasta. Okay, so what about proteins? Proteins are really important because they provide those amino acids. Remember, there's 21 of them. We can produce 12. We get 9 of them from our food. So we make 12. Food gives us the other 9. And amino acids are really important because proteins do basically everything in our body. They're the little workers that accomplish everything. They're also the enzymes that help break everything down. They can come in different structures. So there's the 4 structures, primary through quaternary, and as they get more complex, they can do more things for our body. And some examples of proteins include meat, fish, eggs, nuts. But yeah, meat, fish, eggs, nuts, protein, amino acids, really important. All right, lipids. So these are fats. These are important for insulation. So that's like keeping you warm. That's why seals and sea lions have blubber, because that's just a form of fat that keeps you really warm. They're good for long-lasting energy because our body will actually use fat to store energy. So we'll also say energy storage. Remember, if you eat a bunch of food, your body uses that food for energy. But if you don't use all of that food energy in a day, or over time, your body will start to store it as fat so that it can burn it for later if you need to. So that's why your body will store fat. Other things that fat can do, it helps in our brain. So it helps transmit cell signals. And actually that reminds me of another thing that protein does, can be hormones and communication as well. Okay, so what are some examples of lipids? Well, anything that's like a fat, so like butter, oil, things that are like really greasy, like French fries will have fat in them, chips will have fat in them, but also there's healthy fats like fish oil, nuts, avocado. Those are all healthy fats too. And then getting into butter and oil, remember there's a difference between saturated and unsaturated fat. Saturated fat is straight, which means that they can kind of all pile up on top of each other and block our blood vessels. Meanwhile, unsaturated fat is kinked, which means that they can't really block very well. There's always going to be some pockets that will allow blood cells to move through our blood vessels. So that's why unsaturated tends to be healthier. And it's also room temperature, it's a liquid, versus saturated fat, which is solid at room temperature. So most oils, like olive oil, is unsaturated. Meanwhile, butter is saturated, right? Because butter's a stick of butter. Oil is more liquidy. Okay, so then finally getting to nucleic acids here. Nucleic acids store genetic information. So it's genetic material. It's kind of like the recipe to make us. to make a human, or to make a mammal, to kind of make anything, to make us us. So they come in two different forms, DNA and RNA, and these have two different shapes. DNA is double-stranded, RNA is single-stranded. DNA has the pairs TAGC, while RNA has UAGC, and we'll talk more about that when we get into DNA and RNA. But otherwise, that's pretty much the main thing that we need to know, genetic material that really allows us to be us. And the DNA and RNA essentially then code for building proteins, or amino acids. So this is kind of a just general overview of those four different major macromolecules: carbohydrates, proteins, lipids, and nucleic acids. As long as you can tell me the main function of them and a few examples of those, then you'll be good to go. So that's kind of the end of the chemistry of life. Now let's move into the most recent topic that we're working on, cell structure and function. So first question here, what are three features of a eukaryotic cell, and what are three features of a prokaryotic cell? So let's go ahead and just make a list here. Prokaryote and eukaryote. Perfect. So this will allow me to answer kind of the first four questions here just in this. Let's do a Venn diagram. We haven't drawn a Venn diagram in class yet, but they're really helpful. So we'll do prokaryote and eukaryote. These are not perfect circles, and that's okay. So let's just start naming some traits that they have. Let's start with what they have in common. So they all use ribosomes. They all have a cell membrane. And they all have DNA as their genetic material. So those are kind of those big three main similarities, is that they have DNA, they have a cell membrane, and they have ribosomes. And we've learned now a little bit about what all these things do in the cell. So hopefully that gives you some more context. And once again, they're not just things to memorize, but you can kind of relate them to other things. So now let's kind of look at some differences. So these are some similarities. So let's say what we know about prokaryotes and eukaryotes. Starting with prokaryotes. Their DNA is just kind of free-floating. They tend to be simple. They tend to be smaller. They tend to be, they're not always, but for the most part they tend to be. Let's see, what are some other things? These include bacteria and archaea. Remember, archaea are like the things that live in really intense environments, like really hot, really cold, things that most life can't really live well in. So it's archaea, it's archaic. It's crazy. Let's see, what else? They were the earliest life forms. So there's quite a few things about prokaryotes. Their DNA is kind of free-floating around inside of them. They're simple, smaller. They are made of bacteria and archaea. They're the earliest life forms. Some of them will have cell walls. Some of them will use like flagella or cili to move around, but that's primarily the main things that we've got to do about prokaryotes. Now let's look at eukaryotes. So eukaryotes are different. So unlike prokaryotes, their DNA is in a nucleus, and that's kind of the main thing there. That's the main difference between prokaryotes and eukaryotes, is that eukaryotes have their DNA in a nucleus. They tend to be larger and more complex, more complicated. They can specialize into different things like different tissues and organs, etc., based off of what kind of functions they want to have. Unicellular like an amoeba, which is essentially just one big living thing that has all of its DNA in a nucleus, or they can be multicellular, like us. Multicellular, so that would be like plants, animals, humans, bugs, anything that has multiple cells. So that's eukaryotes. Prokaryotes, DNA free-floating. Eukaryotes, DNA is a nucleus. They all have other fun things about them, but the main three things they have in common: ribosomes, cell membrane, and DNA as their genetic material. All right, so last bit of this study session here, we're going to go through how cells get things in and out of them. So we're just going to make a table. We're going to do passive. You have this table in your notes. Active. And then we'll put up here if they use ATP, if they use a protein channel, and their direction of flow. Okay. So, one of the most passive forms of cell transfer is just diffusion. And remember, diffusion is just the way that molecules naturally will travel. They go high to low. And diffusion is capable of going across the cell membrane if they are small molecules and they're non-charged. Molecules can use simple diffusion to cross the cell membrane. So they don't have to use ATP, and they don't have to use a protein channel because they're small and they're non-charged and they're just doing simple diffusion. So what happens if we have a larger or charged molecule? That's where they'll use facilitated diffusion or facilitated transport. That's because they need the facilitating. They need the help. So this is larger and charged and or charged molecules cross membrane with the help of a protein channel. So they do not use ATP because they're still following just classic diffusion, but because they're large or charged, they do need the help of a protein channel. And because, once again, they're following diffusion, they go high to low. And then finally, one last example of passive transport is osmosis. Remember, this is water. This is just the diffusion of water across the cell membrane. And they also need help. They use aquaporins. And remember, an aquaporin, pore meaning hole, aqua meaning water, they're a hole or a membrane protein for water to move through. Sorry, I keep having to log back into my computer. So they still don't use ATP, but they do use a protein channel, and remember that's the aquaporin. But they still follow the basic rules of diffusion, so they go from high to low. Okay, so that's passive transport. Now let's look at active transport. Active because we use energy, we use ATP. So we've got protein pumps, which essentially just pump the whatever substance it is that needs to be pumped across the membrane. So the pumps molecules in and out of the cell. And remember, the thing that's nice about using a protein pump is they don't have to follow the rules of diffusion, but they do have to use ATP. So they do use ATP, and yes, they do need the protein pumps. So they do need the help of a protein and they go in either direction. They can go from low to high, or they can go from high to low. That's the benefit of having the pump and using energy, is that you have control over the direction. So if the cell wants a lot of something or none of something, they can do that instead of having to deal with having a balance of it, if that makes sense. So that's that. And then we've got those two forms of bulk transport. This is like if you want to do like a few molecules. If you want to do a lot of molecules, you'll do bulk transport. And that's why like people go to Costco, because they can buy in bulk. You can get a lot at one time. And so bulk transport has two different forms: endocytosis and exocytosis. So I think of endo is in, so it's like into, so into the cell. And exo is out of the cell. Exo, exit. And essentially what happens is a vesicle forms around the substance to bring it in or to take it out. And then when it's done, it will fuse back with the cell membrane. And so, do endocytosis and exocytosis use ATP? Yes, they do, because it takes a lot of energy to form a vesicle. And so they use that. Do they use protein channels? No, they use vesicles. So vesicles, no. And then finally, the direction of flow, they can do both. They don't really care what's going on outside of the cell, because they just swallow big old chunks to put in and to spit out of the cell. So this is basically everything that you'll need to know for the final. Of course, there's other review videos that are on Schoology that you can find from the last few tests that go into more detail. So I recommend watching through those if you'd like some extra review. Otherwise, going through the study guides, going through all of your old worksheets, going through all of the notes and slides that you guys use in class. Those are all great ways to prepare for the final. And of course, email me if you have any questions. Otherwise, good luck.